past 2 to 3 years have actually are clearly not as good as we thought initially, or some people thought initially. So we should be more skeptical about some of these progresses that have been made. And also, I think it really speaks to the importance of doing studies and looking at data post-approval, for years, because a lot of this stuff is not that we do. A lot of these treatments aren't really terribly, they're variable in terms of the longevity of the effect. And that's true of all these diseases that are being studied. Everything you read in the paper, oh, this new disease treatment and this, this is not that they do a trial, usually big phase 3 trials in most diseases, but there's no data on how long-term and valid these things are. So I think that's an important point. So I think going forward, I think we have to do more of that, but people are looking at that much more carefully. It's just the initial studies sort of say, oh, this is a miracle. And they show you a video of a kid, walking from the grave, and then, nothing comes of it, or nothing comes of it, but it's not as good as we think, and it's not as long lasting. So I don't have any financial disclosures. I was going to make a joke, but I'm not sure it'll be appreciated, saying, well, I don't get paid enough if that's a financial disclosure, but I'm not sure that is. But otherwise, I have no financial disclosures. A way of getting a higher salary, I guess, by saying that. So the objectives of this talk are to understand the successes in clinical trials of rare childhood neurological diseases, and to understand the pitfalls in such clinical trials. So when I used to review a lot of studies at the NIH back in the day, here, let me get, well, I'll talk about that, but basically, I never thought we'd be doing studies 20, 25 years ago in rare diseases and actually getting approval for treatments. I didn't think it was going to happen, and I'll tell you why in a second. But the agenda here is to compare pediatric to adult trials, discuss some of the approaches for both acute and chronic disease trials in kids, and discuss recent successes. So clinical trials are hard, and they need to be practical. They need to be doable, and you have to have rational and helpful outcome measures. Biomarkers can be very important if they're good biomarkers, because it's hard to do studies looking at an ultimate outcome of perfection, especially when you don't have a lot of patients. Now, so who knew clinical trials in healthcare would be so hard? And I paraphrased Donald J. Trump in 2017, because he said, healthcare is really hard, and who knew it would be so hard? And it's pretty hard. But it's actually kind of amazing that we're getting some of these trials done and getting drugs approved. So in adult trials, it's a little bit easier, because there are a larger number of patients, there are more common diseases, and there are mixed etiologies, like dementia trials. I think that came up a lot of stuff you just read in the news, right? So one thing, there are all these new medications for weight loss. The problem is, and they work really well in large phase 3 trials, because there are a lot of people that are obese that can take these medications, but no one's really looked at how long lasting the effects are of weight loss in these medications. No one's looked at it. So, that's an important thing to do. So clinical diseases are cleaner in regard to pathophysiology in kids compared to adults. Usually there's one disease, it's a mutation, blah, blah, where adults can have more things. But diseases are really less common, so it's very hard to do studies. So the Belmont Report in 1979 summarized ethical principles for human research. And there has to be respect for persons protecting autonomy. Beneficence, a hard word to say, do no harm and minimize risk and maximize benefits. And justice. Kids with rare diseases need justice, fairness, and research. And I never thought they would get it, honestly, because everybody was stuck on these large trials. And I did not think we would get rare disease treatments so quickly. And it was done within 15, 20 years. I'm shocked, actually. So autonomy is a consent. And so here's an inscription on an ancient Egyptian tomb. And it says, this must be an ancient consent form for an experimental mummification process. Maybe they did trials on that. I don't know. They may have. And beneficence, the word I can't say, is there's a hierarchy. One is not to prevent harm. 2 is to remove harm. And 3 is to promote patients' welfare. Children are very vulnerable and require special protections as research subjects. And there is an assent for kids, I think down to 8, you have to have one. Maybe it's a little 10, I don't know. I think it's 8, as I recall, in addition to a consent from the parents. Now, you all remember, well, you probably don't remember because this was in the 60s, I think, and 50s, but thalidomide was a drug treatment and it's lessons for drug safety and regulation. The mothers that took thalidomide had babies that had abnormalities in their arms. This is a kid probably in the 60s who mother had taken thalidomide. I recall it was I think it was an anti-nausea medication for pregnancy. I could be wrong, but that's what I recall now. And they did trials in mothers, and the babies came out deformed. Now, very high rates. So research should not disfavor people on the basis of gender, race, class, age, socioeconomics, religion, or sexual orientation, unless that characteristic is a fundamental variable in an otherwise scientifically and ethically valid investigation. So basically, when the NIH actually tries to do this, but doesn't do it very well. So if there's a study done, let's say, I don't know, Alzheimer's, you're not going to do in kids because basically doesn't exist. But if you do a trial like an adult stroke, there has to be a pediatric component, unless the characteristic is fundamentally variables. Unless there's something that's really primary to adults. What happens practically? They're not pediatric add-ons to these cases, to these studies, for the most part. But it's supposed to happen. So we are supposed to advocate for this, and believe it or not, it doesn't usually happen. They're pretty good about socioeconomics, religion, or sexual orientation. I think kids still get screwed more than any other group. So it's our job to fight for them because no one else is going to. And this is a complex thing. It really is. So the gold standard, which I was raised with, and you guys are taught, is a phase 3 double-blind crossover design powered for significance. Okay, so it has an endpoint, and it's powered to show that that endpoint is met. And that's when if you do a drug trial, you use a drug, double-blind crossover, and it's powered for 90-95% significance, 95% usually. And if you show the drug worked for some metric, it's approved. Okay. The problem is with these small rare diseases, you don't have enough patients to prove significance under these kinds of conditions. So how do we do trials in kids in rare diseases? Well, they're hard. One is acute trials with pediatric stroke, and the other is like chronic trials, and spinal muscular atrophy is the best model, I think. So what happens in pediatric stroke? Well, as you guys probably know, no one's ever done a trial in aspirin prevention of stroke and sickle cell. It will never be done now. But there was a time when it should have been done, never done. And there are reasons for that. Some of them they're not really political, but hematologists generally don't like aspirin. Neurologists love aspirin. So a trial was never done. And they would try to power them at, phase 3 levels, and it fell apart. Another trial that was tried, it wasn't such a large trial, but was a trial for TPA in kids. Now, as you know, kids don't usually come in within 3 hours of a stroke, but some do. And what they did, the trial was very terribly designed by good people, right? So what happened is they had so many inclusion and exclusion criteria that you couldn't get enough patients. So you had to show that there was a clot on an MRA before you could give TPA, which you had to do within 3 hours. So guess what happened? No patients were enrolled, but you could have guessed that before you did the study. So you have to be very careful in what your inclusion, exclusion criteria are, especially in kids. Now, in adult stroke, what they basically did, as I remember the studies, they just said, patient had a clinical stroke, CT scan doesn't show hemorrhage, you gave TPA, and that's how they did the study. And we could do that in kids, but we're never brave enough to do it. And the reason in kids, it's harder because everybody said, well, there are more migraines or seizures. And the answer is, well, so what? TPA is probably pretty safe. And it should have been much more open-ended to do it. But everybody was too scared. They wanted to show there was a clot first, and nothing happened. So we don't have a trial in TPA, and probably never will. But what's happening is people are giving TPA in kids. And we finally did one, I think it was a 17-year-old at King's, got TPA, I guess, last week. So that's pretty cool. And it is happening. The kid had a stroke in hospital, which is always easier. But there's never going to be a trial. And there's never going to be a trial of aspirin and sickle cell. And, it just isn't going to happen. So a lot of pediatric research clinical trials start with disease registries. So you have to have collaborators, and the collaborators have to have benefits and buy-ins. So they have to be on the paper, for example. You have to give them money, but if you have grants, you give them money, or you put them on the papers, and they get benefits and they buy into the study. And that's a good way to start. And that's been done with a lot of different pediatric strokes. With pediatric strokes, we've done multiple sclerosis, sickle cell disease, spinal muscular atrophy, muscular dystrophy, rare disease associated with mitochondrial disease. Family groups are sometimes helpful. Sometimes that gets very political, and sudden unexplained death and epilepsy syndrome. There are collaborative groups that look at these things, and that's how we get data. And it does work. I guess sickle cell was really early. Sickle cell disease has been studied collaboratively since the, I guess, late 70s. I got involved in the 80s. And what the government did, sometimes the government does great things. At that time, it set up 10, then became 11 sickle cell centers that were funded. So they just gave grants, and you could do anything you want within that the project grant. And most of the stuff was kind of lousy. They had, social workers getting the kids to clinics and stuff, which is fine, but it's not going to cure a disease, certainly. Same thing happened with AIDS at the beginning. That's basically what they were funding. But sickle cell disease, a lot of studies came out in those collaborative groups within institutions, and it worked. We basically solved most stroke in sickle cell. It's kind of gone now, both preventative and secondary prevention. But primary prevention, which is a big deal. We have primary prevention in sickle cell, and now hydroxyurea seems to decrease the risk of stroke anyway, which was never really studied primarily, but it has been given so much by hematologists we can look at the data. But the STOP study, which showed that TCD, elevated velocities of TCD, are associated with a stroke soon after, resulted in transfusions starting with critical TCD values and resulted in primary prevention of pediatric sickle cell disease stroke. And again, it's not so common now because I think hydroxyurea has made a huge difference. Acute trials engage patient groups and physicians early, crucial. Multiple investigators need to collaborate. Be sure, and this is very important, be sure that everybody has skin in the game and reaps benefits from collaboration. This is not hard, but politics often rears an ugly head. There's always a couple of people that sort of take over the thing. Examples are IPSS, International Pediatric Strokes, which was very successful, but did have internal politics that caused problems. MS, sickle cell groups, use approved drugs that are generic results in easier access to the drug, easier treatment trials, and safer, even though they're used off-label. So in acute trials, you really have to have novel designs, especially in rare diseases. So if you do a phase 1 trial, it's potentially effective. You go to a study that's open label and you do it versus historical controls. Now, our generation was traditionally taught that historical controls aren't very good. There's a paper in the New England Journal showing that they don't work. But guess what? We have to do it because there is no choice. And everybody's sort of forgotten that, but it's better than nothing. And if we don't loosen up our strong beliefs, it's gonna be a problem. Phase 1 dose increases if potentially effective, and then you do the randomizing crossover. So you go from one to the other, boom, boom, boom. You don't sit around and try to get other grants and studies. You sort of set it up this way. Now, this is an interesting study that still hasn't happened. So we were talking, and we're going to be part of this, but this is doing comparative effectiveness designs, doing really weird designs for trials that you couldn't do normally. So there's a focal cerebral arteriopathy study, which I think is funded, but the money's not been given. But we're going to do it probably in all 3 institutions here. So we'll study steroids in our focal arteriopathy in kids with strokes. So that's very rare. I think amongst our 3 institutions, we might see 1 kid every 2 to 3 years. So probably in 5 years, we'll have 2 or 3 kids. And so it's pretty rare. And there are a lot of these kids probably in big community hospitals, but they're never going to be studied because it takes too much trouble. And there's not enough money in. So what we're going to do is treat them with steroids. And I'll tell you why we would do that in a second. But you need lots of buy-in from investigators. And there's no need for a control arm, which was typically standard in phase 3 trials. You had to have a control arm. This is embedded in a natural history study, okay? i.e. alone or with steroids. And the steroids are 2 arms. Now, let me tell you where that comes from, just so you know. In the 60s and 70s, well, in the 60s, 70s actually, pediatric stroke was thought to be dissection all the time by adult stroke people. So that's what they wrote. And then what happened is most of the pediatric neurologists that were interested in stroke thought it was inflammatory. Same angiograms. just so you know. And so they would give steroids in the 60s and 70s. A patient had a stroke with an arteriopathy would put on prednisone. And that was a standard of care. And then, but people sort of forgot that and went away because we started comparing ourselves to adult studies, and we gave everybody first heparin, which is not being done now, and then TPA, which is going to start, and has started. So that's where this trial comes in. Now, the comparative effectiveness design is when the patient comes in with arteriopathy and a stroke, there are basically two arms. Well, there'll be three arms. Patients are, if the doctors don't put them on steroids, we'll just look at aspirin outcomes. And certain centers won't do it, for example, but we'll look at their data. So if Maimonides doesn't want to give steroids, they'll say, fine, we'll just put the patients in the trial with aspirin and see what they do. Not perfect, but better than nothing. Okay. I guess the story here is better than nothing is better than nothing, right? And the other two arms are patients come in, they're put on steroids, and the crossover design is the other group will not be put on steroids, so it's just put on aspirin, which is a standard of care, even though that's not been studied. No trials for aspirin in this situation in kids. And that arm will follow MRAs. If the MRA worsens, steroids will be started. So there are two, it's a comparative design, and patients not treated with steroids will be looked at. So there'll be that'll be another arm, but not a standard phase 3 trial, because this is really rare stuff. And we'll see what happens. We're supposed to start, but I'm not sure where the funding is, if there's a problem with budgets or not right now. I'm running the grant, we're part of it. I helped set it up, but now we're just being part of it. So sometimes in relatively rare disease, you can do phase 3 trials. So in spinal muscular atrophy, nusinersen was proved effective in SMA, and steroid trial. This was a big trial done in DMD. Are effective in increasing walking, and those were done in large phase 3 trials. So it can happen. There's noise out there. Someone's getting in. So SMA nusinersen is an antisense oligonucleotide designed to treat patients who have diseases caused by mutations in chromosome 5q that lead to SMN protein deficiency. This was FDA approved, I guess it was 2018. Now, I remember the first videos that we saw of these patients, right? There was a video out of Columbus, Ohio, and National Children's, and someone infused the nusinersen, and the patient walked up. It was like miraculous. It was like Lazarus walking up from the dead. Now, you have to realize, when I was starting out, SMA was lethal, okay? And I used to, when I met a baby with SMA, for example, what I used to do, and everybody didn't do this, but I would talk about DNR, whether the parents wanted it or not. Because what would happen in 6 months, the patient would show up in the emergency room and the parents didn't know what they wanted, they'd be intubated. So I would have a discussion by the second visit. And I offered nothing else other than that, which is kind of lousy, but that's what I remember. Okay. I once got a call. There was a patient of one of my colleagues at Columbia. The kid was being admitted, being intubated at Penn, at CHOP, and then the residents didn't want to do it. So they called me at 2 a.m. because they couldn't get a hold of my colleague. And they said, what do we do? And I said, you got to, if this has not been discussed, you can discuss with the parents now, but you have to intubate if there's no one saying it, talking about it. So that was that was a level of what we were doing. And I remember that call at 2 a.m. going like, oh my goodness, this is ridiculous. And my colleague never talked about this too, I can tell you that. And I won't mention who it is, but that would irritate me. So SMA gene therapy has come about now and has been approved by the FDA. What is interesting, when I first gave this talk, we knew that there were patients being treated with both nusinersen and gene therapy. So neither worked that well. They might get a gene therapy, then get nusinersen too. So we kind of knew these weren't totally perfect treatments. But it's become clearer that there are other issues with it. And for example, why is gene therapy not always totally effective? There's a lot of variability between patient treatments. So why is that? Well, one could be delivery, right? Two, the gene could sort of be replaced by the abnormal gene with time. And there might be other modifying factors in the patient that affect the disease. So these are not miracle drugs, but they're pretty good. We're still talking about patients with infantile spinal muscular atrophy not dying. But clearly the treatment, especially for gene therapy, what I should mention should be as early as possible. Sometimes the train has left the station in these medications. And I think that's true in sickle cell. Sometimes there's so much damage to tissue and endothelium and everything else that it's too late to get rid of it, to fix the gene. The disease might just go on its merry way. So I think we have to be a lot more careful. A lot of these design studies are done by drug companies. They want to show effect and they want to sell their product. Okay. So when the SMA gene therapy came out, it's $2 million for one treatment. But the drug company said, oh, I think it's Novartis, I don't remember. I'm sure one of you guys knows. But and the drug companies are great because this stuff wouldn't happen without drug companies. It's amazing. But they sold this drug and saying it's a one-time shot and the patient's going to be cured. Well, it's not true. It doesn't cure everybody. And there are, when we started seeing patients on two of these drugs, we knew they were infected. Now, for Duchenne muscular dystrophy, the standard is steroids, and that was a phase 3 trial. And now they're doing another trial, which I think it has been published, I think. I don't know if it's been approved, of another type of steroid that may have less side effects. And it's a big phase 3 trial, and it's fine. But we don't know how well it's going to work, if it's going to really have less side effects. And it's more expensive, and the drug company is selling it. And it's easy for a drug company to do a phase 3 trial in the form of steroids. And the exon skipping has been approved. I'm not sure if these other two therapies have been approved because I didn't look it up again, but they weren't approved in 2019. Now, the other thing you can do statistical innovation, which I don't fully understand, but I'll tell you what it is, as far as I understand it. Bayesian methods are characterized as follows. The use of more generally unknown quantities to model all sources of uncertainty in statistical models, including uncertainty resulting from lack of information. So in a clinical trial, traditionally, statistics were basically, okay, this is the outcome that we're going to look at. And we're going to study the patients in 2 arms crossed over. And if another outcome comes up, we can't use it because that wasn't designed that way. Trial's finished. But this is the idea, you have to be much more flexible. And then need to determine the prior probability of distribution, taking into account the available information. So that's how you start. But you don't have all the available information, so one cannot be too rigid. When more data becomes available, calculate the posterior distribution. Subsequently, the posterior distribution becomes the next prior, I guess that's the right word. Basically, you have to adapt your statistics. And if you don't, you're very rigid and a lot of things are going to fail. Okay. I remember when I was I was on an NIH study section for it seems like half my life. And I remember asking the NIH, one of the NIH people, and that they had, a guy comes up and presents the study that shows that we're doing much better with the grants at the NIH because they're starting within, let's say, 4 months. And this is a great thing, big breakthrough, kids. And they had slides and stuff like that. So I raised my hand because I'm oppositional. I think that's why they liked me at the time, but maybe not anymore. And I say, well, how often, for example, if it's a drug trial, do we find efficaciousness? And no data on that. Too hard to look at me. And that's an important question because maybe our trials kind of aren't very good if we don't find it. And my sense is, I never could get that data, is that most clinical drug trials run by the NIH did not work for the reasons we stated. They were too rigid, statistics were rigid, people, these committees would have who's going to be coming to the study and inclusion, exclusion criteria. They were so rigid, you couldn't do it. They're doing construction outside. I'm sorry if it's noisy. So other trial innovations, carefully add investigators and discuss equipoise. Now, equipoise is a term that everybody should know. So I'm going to define it for you, but most of you guys do know, I think. But equipoise means that there is an agreement, is what the agreement to do the trial. So if everybody believes aspirin works really well and won't use steroids, you can't do the trial. There's no equipoise. But if some people don't believe aspirin's 100%, then you can do the trial. You got to, as we said, develop practical and doable inclusion criteria. You repeat measures over time. You do additional interim analyses, which are not done very much with phase 3 trials. Now, dropouts provide data too. Never give up on any data. And this is something that people don't do. And for treatment trials, use models of drugs already in use if you can. Doesn't mean you have to do that, but it makes it a lot easier. So dropouts provide data. And the other thing I want to add is that, which is not on the slide, but after the study ends, you really have to look at the data. What's the durability of giving gene therapy in sickle cell disease? I don't think that's been looked at. And that should have been put into the initial trial. Okay. And what was the patient clinically like when they started the initial therapy? I don't think we know. We just give it to everybody. Oh yeah, it works. Gets rid of the. What's the outcome? Oh, you change the mutation. Oh, does that affect the patient? We don't know. But the drug company doesn't really want to do that because they want to say it affects the mutation. We have a treatment that costs you millions of dollars, and you all should take it now. Okay, so so we as physicians have to really sort of step in. The drug companies are very important because none of this stuff would be done. But on the other hand, they don't really want to know the nuances because it doesn't benefit them. Now, there are platforms that we can use that make people work together. In Europe, it's called the Orphanet and recognizes hundreds of rare diseases and provides data and information. In the US, we have Neuronext, which does trials in neurological diseases in every institution. Institutions that want to do that are part of it. We are part of Neuronext. And in fact, the FOCUS trial is going to go through Neuronext. It's such an NIH funding mechanism. And StrokeNet for strokes. Okay. I guess the FOCUS trial, the steroid trial, could go through StrokeNet, but I think it's going through Neuronext. But I could be wrong. And that's been in the process for three years now. I can't believe we're pulling it off, but it's got to be pulled off. Okay, what is a small sample? Well, there are 63 orphan drugs approved in the EU between 2000 and 2010 with a sample size of less than 50 patients. I would have never believed this would happen. If you told me this in 2005, I didn't know about these orphan drugs being approved in the EU at that time, but I would say it's impossible because everybody's so rigid with trial designs. They're never going to believe it. They're never going to do it. Because I was taught to not, in the traditional phase 3 way. Now, FDA has expedited programs, which we can debate can be good or bad. So Office of New Drugs and Rare Diseases, for example, there's a Morquio enzyme replacement that went through this, allosulfase. There's lots of examples of this that are going through and getting through fast. And there's a compassionate use outside of trials. Now, the other problem that happens, though, I don't follow the dementia stuff that well, but I see it on the news. It looks like these things that are being approved by the FDA have marginal effects. And no one's really looked at longevity of the effect. And what they're looking at is an outcome measure. Most of the studies looked at amyloid, which is not clearly a marker. And anybody who wants to correct me at the end, they can, because I don't know much about this. And the only dementia we have is in Alzheimer's type dementias and Down's kids, and they're usually happening in the 40s. And then they're looking at some other cognitive measures now that are more real world. But the effects don't seem huge. And these drugs are expensive. And again, I'm not going to speak to them in any real rational way, but because I don't know enough to do it, but I'm a little, I question it. Now, this is a list from in 2021 that's listed in the Lancet. So again, the Lancet, just out of interest, was kind of a leftist socialist magazine out of England in the, I guess it's been, I mean, when I was reading it, it was in the 90s, and it's become more of a capitalist paper now, but it's still pretty interesting. So they're looking at these new drugs on the basis of investing in biotech. And investing in biotech is a good thing. I'm not saying you shouldn't, because it's helpful to the world, right? And it's probably not a bad thing to be doing in large numbers. But BioMarin is a company that makes drugs for hemophilia. Now, Bluebird Bio is actually interesting. I don't know, I haven't really looked at all their data, but they have an approved treatment for adrenal leukodystrophy. It's a genetic treatment. So what BioBird does, whether it's better or not, and again, I'm sure we can look at this data, for example, the adrenal leukodystrophy trial was approved with a proviso that they have to look at patients long-term. So what good is it to, I mean, it's sort of like, some of these cancer drugs that give you one more month of survival when you go to sort of like, okay, great. Like, would I take that if they offered me that? No. Like, I don't really, unless I had something to do, like do a will, or I needed more time to do something, but it seems kind of like really. I mean, you can work to make it better, but I'm not sure that's going to happen. So as this was approved as a gene therapy, and what they do, it's a little different than other gene therapy trials. Is they take the patient's stem cells and enter the gene, the wild type gene into that, and then put the stem cells back into the patient. So adrenal leukodystrophy basically was not treated except for using bone marrow transplantation. That was the big thing even 30 years ago. Problem with adrenal leukodystrophy, once the damage is done, that it's probably not terribly reversed. So the best patients to treat, of course, are the ones that are picked up in standard screening of newborns. And that's adrenal leukodystrophy was not screened normally, was not screened in the newborn screenings in most states up until 2018 or so, once people started thinking about this. I think it came out around 2018. So if you give a bone marrow transplant or you give this treatment, that should be done as early as possible. And you'll do better. And their treatments, Bluebird has treatments for sickle cell and beta thalassemia. There may or may not be data out on this, but what I'd be interested in is the durability of these treatment responses. Is it better with a stem cell? You would think delivery is better by doing that. Sort of personalized medicine, right? And you're using the patient's own stem cells. So you would think it would be better, but I don't know if that's true. And I'm not sure there's any data yet. But for adrenal leukodystrophy, I know that the FDA said, you guys got to follow these patients. And the same thing should be done for the treatment of SMA. I mean, how long does that work and in how many patients? You know, it's $2 million. We're not talking about nothing. So there's a drug, Gencyte, for Leber hereditary optic neuropathy, which is more of an adult disease. Kids can get it. There's another drug, I think it's an oligonucleotide drug for SMA by Novartis. There's a treatment for metachromatic leukodystrophy, which is an enzyme replacement. And that's a crappy disease. But the last patient I saw with that was about 6 months ago. It was here. And it was classic metachromatic leukodystrophy. And those patients are pretty just, by the time you figure it out, the patients are pretty impaired. I think the kid was real. And I called up one of my friends that does most of these studies on this, and it was really, it would only have been like put him on a trial. And for some immune deficiencies, there are medications that work for them. So these are approved medications in some jurisdiction in the world. And this is as of August 17th, 2022. So they had a table of this. It's pretty cool. In The Economist. So why don't gene therapies always work? Well, again, I think I've talked about it. Delivery is a problem. And I'm kind of thinking stem cell sounds good, but I don't know that it's any better. There might be a loss of the active gene with time, sustainability. And I think the timing of treatment is very important. And from what I've seen, and you guys can correct me if you know, that people haven't really looked at that, carefully. It's just that let's give the gene for wild type, get rid of sickle cell mutation. And if it works, you look at the mutation, it's gone. Hey, Eureka, that's great. Give me $1 million, I'll give it to all your kids. And I'm not sure that's good. There were studies in the past, small studies, that showed that kids with sickle cell and arteriopathy sometimes worsened even with transfusion. So justice in child trials of rare diseases, it's happening, and I am very overwhelmingly happy about it. It requires flexibility in design, inclusion criteria, statistical methods, frequent interim analysis, and no rigid thinking. Focus trials are a great example of that, where you have two arms, one steroids right away, and the other steroids if the patient gets worse. And incidentally, I do think steroids work in that condition. I don't know why, but I think they do. So the future. Now, the difference between this talk now, and again, I haven't really upgraded it very much, so I'm sorry. Even the slides are old-fashioned downstate. But I didn't, I said, okay, I'll do it last night. It's going to be more of the same. And it's very impressive. Strides in the field have been heartening. And I find this is impressive, but not as good as we thought initially when they showed slides, videos of kids just jumping up with SMA and walking and dancing. That's what I remember there was this video going around about this kid with SMA getting Nucerin, and it was like, oh my goodness, I can't believe this. And this comes to a disease that we thought was uniformly fatal. Earth-shattering. So in my lifetime, I've seen this stuff. I guess it's like my father saw the first cars come out. It must have been amazing. It must have been that same kind of level, right? Now, I find bureaucracy worse and worse in terms of studies. And we're making breakthroughs and breaking through the bureaucracies, and I think in a way that's quite cool. Okay. So we got to save babies. When I gave this talk, pediatric neurologists are sort of self-deprecating and feel badly about themselves. We were looked down upon by adult neurologists and the rest of the world. So we really are insecure. So I gave this talk, and my partner gave me, who's in acting and stuff in comedy, says, you got to, any comedian would come into the city and say, rather than saying, thank you to the committee for inviting me to come, which is pretty boring. And that's what everybody else said, to give me a plenary talk. She said, where else in the world can you go a block from your apartment and get the best spondycopia in the world for €2? So I did that, and everybody loved that. And then I did this thing about saving babies. And I think we should feel better about ourselves, because sometimes we do feel like we're secondary. So we're secondary to adult neurology, we're secondary to pediatrics, we're not in the departments, and that's part of our burden that we have. But we do save babies, and we try this every day. Not always. Unfortunately, I saw a kid that has a genetic mutation yesterday that it's life-threatening, and we're not going to be able to fix it. But we can try to help the people. Isabel Rappin used to, and what we do is kind of a calling, even though the science is amazing. And she used to say it was a privilege to take care of patients. I've quoted her a lot in the past couple of days. I don't know why. She died about 10 years ago, and she was an attending at Einstein. And she was Jewish, but very Germanic, because she was from Switzerland. And I found her very kind of like, oh my goodness. But she was great. And the more you get to know her, the more she loosened up a little bit. And she was a great pediatric neurologist. And she was the one that really sort of set the stage for saying autism is more common and defining it differently. So since I gave it in Greece, and back then it was even more, I thought the treatments were even better than they are now. Now we're seeing all the problems with them. That's okay. We'd expect that. I was watching something on TV. I get all my science information now from lay stuff, which is kind of cool. And they were talking about these these diabetes type 2 medications, the injectables, and they really work. They work really, really well. I mean, people lose 15% of their weight in 4 months, or 20% or huge amounts of weight, and they really work. The problem is there's no data, and these drugs are very expensive. There's absolutely no data on the durability of those treatments. So some people have been reported to gain twice as much weight back when they stop. So sounds good, but maybe not so great. And they can do phase 3 trials on this, but no one's really looked at it. So I had this slide in my last slide. So since we're in Greece, let's dance. Even though I don't like dancing, but I guess the Greeks do. So this is a tape from Zorba the Greek, which is a very dark film. I looked at it again during COVID. I hadn't looked at it in many years. It was like the early 60s. And Zorba is a very, very dark film. And Zorba himself in the book was kind of a misogynist. My daughter said, you shouldn't show it. She says, doesn't matter. I'm not saying he's a good person. I'm just showing the film. So since we're in Greece and things are going so well, and I have to tell you, I am actually in awe of what the progress we're making. But, it's usually not as good as it seems initially. That's what we always have to be. And there are other studies to do. I showed this clip, which is the end scene in Zorba the Greek by, and here's Anthony Quinn. And so this is good. Now, I can't dance at all, so, it's not a genetic thing. So this is in black and white. You should all look at this film. It's very dark. And that's Alan Bates. He's an English actor. But they're so happy that we did these trials. Amazing. But remember, in '63, we knew nothing about anything. So it's amazing. It is actually amazing. Crete's really nice too. So if you can go there sometime, it's nice. Okay, so that's it. And I will take comments, questions, or corrections, because I don't know this field perfectly, especially stuff with the dementia stuff, the Alzheimer's stuff. I haven't looked at those trials carefully, but on the surface, I'm kind of worried about them. Okay, any questions or comments or complaints? Again, be nice to me, because I just did this last minute. And this is an old talk.
Thank you so much, Dr. Pavlakis. It was an amazing talk. So does anybody have any questions?
Steve has 2 comments. One, you're overpaid.
Oh, I knew you would not like that, but I didn't know what financial disclosure was. The only thing I had is I'm underpaid, so that's no one's ever said that as a joke, but I figured I'd try.
And number 2, I think you underestimated or did not really give Isabel the accolades she deserved. She was an absolute giant.
Well, she was. Yeah, and I've been talking about her lately for a few days. I spent 2 days with her on setting up some NIH stuff for AIDS, and it was great. The first day she kept looking at me, and then the second day she laughed the whole time. She had a good time. So she was a giant, and I like her, and I've quoted her a lot in the past few days. I've mentioned her a lot, and I think saying that this is a privilege to take care of patients is a great quote.
Yeah.
So 2 complaints you had. You didn't say anything good about me. Jeez.
I don't want that getting to your head, Steve. How wonderful.
That's not going to, trust me. Anybody else have a question or comment or?
Yeah, Steve, it's Brian and Jessica. The enormous triumph with therapy for AIDS has been one of the most amazing phenomena I've seen in medicine ever.
Yes, there's some patients cured now of AIDS.
I mean, yeah, but the fact that it was, I remember the 1980, at one of the first grand rounds given by someone from NYU who described a new disease in homosexual males in 1981. The fact that the lifespan is almost normal, and that it's certainly not transmissible in many people who are well treated, is an astonishing phenomenon.
It was astonishing.
And I think that whenever people do trials or look at new things or old diseases, they should look at this amazing, I think, one of the greatest accomplishments American history has ever made.
I think so too.
In science. I just wanted, people aren't exactly aware of how much progress was made. And really thanks to Dr. Fauci, who's not a researcher himself, but who listened to people and moved ahead with the research so that by the mid-1990s, we had effective treatment for what was uniformly a fatal disease. I remember working at Kings County and people just dying like flies, young men usually.
Yeah, no, I totally agree. I remember in '88, we're sitting around talking in someone's office and we said, we don't know how to treat viruses. And now we kind of are starting to, right? We can do that with COVID a little bit. We can do that with flu a bit. And it was phenomenal. Now, as I remember, because I was on some grants, but what the government did there was a little bit like sickle cell. There was so much push to do something clinically, and there were a lot of money thrown at doing clinical things that didn't really change anything. The basic science changed the treatment of AIDS, and it was one of the phenomenal things in our careers. And now it's even, we're just doing it all over the place, and it's not just us. So I guess you think Fauci should not be impeached. Is that what you're saying?
No, of course. I mean, I think that the country owes enormous debt of gratitude to him.
Yes, I agree. And what happened with AIDS was in my career was probably one of the first phenomenal changes in healthcare, and did change everything. And it was quite impressive. I remember that we said, we don't know how to treat viruses. We can do bacteria pretty well, but we have no idea how to treat a virus. And now we kind of do in some things. Right? Anything else? So that's why we dance, because this is so amazing. But I think the AIDS is the major. AZT was the first drug. I'm pretty sure it was.
Yes.
And that was phenomenal. And then we gave it AZT to mothers, and the babies did not, I think it was 1 in 5 babies had HIV, the mother with AIDS, and it stopped it. It was never transmitted if the mother was on AZT. So anyway, pretty cool stuff. So that's why it's great what we do, and it's a privilege to be a doctor now. Even if some of us are underpaid. We do it for the joy and the privilege. All right, anything else? And otherwise I'll shut it down, and I'm a little early, but that's okay. So maybe I'll fix this talk. Go ahead.
This is a kind of comment on medicine in general. While what you show today and what has been done is really amazing since the 1960s and 70s. Nevertheless, the medical profession is under huge attack on abortions, on end-of-life care, on drugs, etc. We seem to be in everybody's, gun sights. Yet we've never been as successful as we have been now. We certainly have a long way to go still. But we should be aware of defending the medical profession against constant attacks and attempts to undermine us.
Yeah, I think it comes from both sides of the political spectrum. There's this, Ida and Alex and I were talking about there's a SUDEP thing that we're supposed to do in New York State, and there's no real penalty if you don't do it the first time, so it's fine. But basically saying that if you don't tell someone that has a seizure, even one seizure, looks like it's great reads, that SUDEP exists. And you have to tell them in a culturally appropriate and a language kind of way, which is totally ridiculous. I mean, it's totally taking the the state house and the governor are basically telling us what to do about medical care. And same thing with abortions. That's ridiculous. And these are things you do individually and with a family or not. But we are under attack, and I think it's absolutely absurd. All right. So we should fight and fight for kids. Forget old people; they're already going to die soon anyway. Who cares about Alzheimer's? Let them just go. Go to the nursing home. Let them go. Just treat the kids. All right. Else? All right. Thank you all, and thanks for your attention. And have a nice weekend. I guess we have a long weekend coming too. Bye-bye.
Thank you, Dr. Pavlakis.